For many years, human diseases have been simulated through in vivo studies with model organisms or in vitro assays. C. elegans is a model organism that has been widely accepted by the scientific community since its introduction in 1973. C. elegans are small nematodic worms that have a very short life cycle, possess full body transparency, and are amenable to extensive genetic manipulation. Due to these advantages, it has been widely used to simulate several human diseases in the past, ranging from diabetes to neurodegeneration, including Parkinson's and Alzheimer's disease. The genetic manipulation allows for expression of disease-specific genes, toxins, or proteins within the worm's body, and the several behaviors it possesses allow for quantification of different phenotypes, providing a platform for drug screening.
Due to the small size of the worm and continuous sinusoidal motion, different immobilization techniques have also been introduced to allow for high-resolution imaging. The conventional chemical immobilization through adhesives has been shown to permanently damage the worm's health, while mechanical constriction in microfluidic devices require more complex fabrication steps. Through previous experiments our lab has conducted in the field, we have identified a new C. elegans behavior that can be used for full-body mobilization and high-resolution imaging of specific parts in the worm. The novel microfluidic technique utilizes direct current electric field application in order to immobilize the worm. The experimental setup consists of a single-channel microfluidic device connected to a microscope for image acquisition and a MATLAB-controlled source meter for the application of electric field.
The microfluidic device was designed through SolidWorks and fabricated through soft lithography on silicon wafers with polydimethylsiloxane or PDMS, a common polymer in microfluidics. The inlet and outlet of the device are punctured with wires to allow for electric field application, and the device is bonded with oxygen plasma onto a glass substrate. The 300 micrometer in width single channel encloses an 80 micrometer wide trap to prevent the worm's escape during the experiment.
The complete immobilization of the worm is observed through the absence of the sinusoidal shape, measured by quantifying the motion of the head of the nematode. The quantification is completed through a custom-made MATLAB code. When the current is applied, the frequency reduces to zero, while when the stimulus is removed, the worm continues its motion to its healthy standards, proving that our technique does not affect the health of the organism. Due to the width variability within the channel, different simulations had to be run onto the microfluidic design in order to determine the uniformity of electric field applied, as well as the voltage that needs to be supplied to achieve the desired exposure. Through COMSOL simulations, it was determined that the electric field within the trap is uniform and that approximately 7 volts need to be applied to achieve complete immobilization in this device. Our previous studies have shown that complete immobilization occurs at electric fields higher than 6 volts per centimeter. Full body mobilization at different magnifications allows for imaging of specific features at high resolution that are important factors for C. elegans health and developmental studies. Using our technique, we have been able to image the pharynx and vulva of the worm at very high magnifications and resolution. We were also able to test the mutant fluorescent strain LX1918, expressing the green fluorescent protein, or GFP, in the hermaphrodite-specific neurons. The 5-second exposure to electric field provides a window of full-body mobilization that allows for high-resolution imaging. The fluorescently tagged neurons, along with the major C. elegans features, are distinguishable, and the fluorescence intensity is quantifiable. In conclusion, we're proposing a novel microfluidic method for full-body mobilization of C. elegans that allows for mobility recovery for the worm. We have shown that our device can immobilize the worm and acquire high-resolution images of in-body features as well as fluorescent structures. The full-body mobilization technique that is presented here can be included in any microfluidic setup, produce a robust behavior from the worms, and allow for high-resolution imaging. Our future work on the project includes drug screening of Parkinson's disease-related chemicals and in specific 6-hydroxydopamine, or 6-HDA, a common neurotoxin for the disease. To improve the throughput of the device, a high-throughput design with multiple channels is currently in development through our lab. Thank you for your attention. Please feel free to contact me if you have any questions.